Hello everyone, I'm Lingfei Wang. I would like to present to you Normalisr, inferring single-cell differential and co-expression with linear association testing. This is based on my work and the Broad Institute. I would like to thank the organizers for giving me the chance to present my work here. I also have a complimentary poster for this talk. So if you start from the bulk area and think about inferring gene regulations from empirical evidence, you typically start from transcriptome data, sometimes also with the genome, to look at questions like differential expression, co-expression, EQTL, and causal inference to look for gene regulations. However, these are merely empirical evidence, but doesn't tell you why and how the gene regulations happen. For that, you need to use other data types like epigenomics. There are major drawbacks in bulk RNA sequencing and transcriptome and these kind of gene regulation inference. For example, many of the studies like co-expression, EQTL, and causal inference, they require quite many samples, like hundreds, for example, and these can be quite costly in the bulk sequencing. There are also limitations on the perturbation side because you can only have one perturbation for one bulk sample. At the same time, even if you find differential expression or co-expression, it's pretty uncertain where they came from. It might be a certain cell type, you don't know which, but it might be just cell type composition changes, but not really changes of gene expression in any of them. These questions can be addressed with single-cell RNA-seq. By regarding each cell as an independent sample, single-cell RNA-seq gets a much stronger statistical power just from the number of cells. At the same time, a single RNA-seq can know each cell's cell type, and therefore you can choose only the cells you're interested in for higher specificity. You can even manipulate cells individually, for example, in a pooled crystal ice cream. However, single-cell RNA-seq has major statistical challenges, mostly known ones are the sparsity and technical confounding. Therefore, we have found difficulty in deriving new methods and analysis for single-cell RNA-seq. For example, for differential expression, a benchmarking paper about 2 years ago found actually the differential expression method designed specifically for single-cell actually doesn't perform much better than Agile, which is what we normally use for bulk RNA-seq. At the same time, single-cell co-expression still haven't really provided a really high-quality co-expression network, especially at gene level, and can provide interesting biological insights for our analysis. Another question would be how to control for false discovery in co-expression, because it will make it possible to compare across data sets and across cell types. In the end, we also hope to unify differential expression and co-expression in the same, hopefully simple framework. And therefore, this would make it possible to do more complicated advanced analysis like EQTL discovery and causal inference. Particularly, it would be a bit difficult to do it with generalized linear model. For example, for differential expression, people typically use negative binomial distribution because the GLM are known to have difficulty generalizing to co-expression patterns. While the efforts of unifying differential and co-expression have been done by many people using normalization and imputation methods, actually. So if you look at here, on top you have the biochemical process, which is actually hidden from the observers and experimentalists. In the beginning, you have some unknown regulatory network that exists within every cell. But for each cell, when you have the mRNA profile, it's only a snapshot of the regulatory network you actually have, and that adds a biological variation on top of it. When you actually do the single-cell RNA sequencing, you then add technical variation for the sampling process. You could have the read count visible, but after that, you do normalization or imputation, hopefully to reach some expression level that estimates the actual cell mRNA profiles as well as possible. Then from the normalized or imputed expression, you can do any kind of analysis you want, like differential expression or co-expression, to infer the gene relationships, hopefully to recover the regulatory network. So the second part, the bottom part half of the figure, of the panel figure here, can actually be observed, and it's what we do for statistical inference. So this is what we thought in the beginning, and that is why we try to use existing methods, for example, MAGIC and DCA for imputation, and even log CPM for normalization. In this case, we compare two scenarios, which is first positive control differential expression and then the negative control co-expression. So in the first case, we use a CRISPR-I perturb-seq to look for whether the targeted gene are differentially expressed or even inhibited by the guide RNAs. After imputation with MAGIC or DCA, we actually don't see a really strong p-value compared to the inference by Agile. If we do it with log CPM normalization, we do find comparable significance or sensitivity in terms of positive control of differential expression. On the other hand, when we look at the negative control of co-expression, because in reality you can't really get real data of no co-expression, we generated a synthetic data set using log normal distribution of the for the biological variation and multinomial distribution for the technical sampling process. In this case, we started without any co-expression, and then we imputed the expressions from the simulated UMI reads with MAGIC or DCA, or we normalized them with log CPM. This turns out to be an even more difficult question, because when you are supposed to see a uniform distribution of p-value, you actually see an enrichment of very small p-values indicating a false positive by all three methods. Well, this is probably also because when the methods like MAGIC and DCA, when they were proposed, they were looking at clustering and visualization questions rather than differential and co-expression questions specifically. that motivated us to design this normalizer method to first normalize the expressions and covariates, and then to perform linear association testing. So you start from a UMI matrix where you have the number of genes as the rows, and the number of cells as the columns here. The first question we asked was, how do we get some like log transformation, but without thinking about log CPM or log CP10K, because we see this million or 10K would be pretty artificial. So the question we considered was, if you have a large pool of balls, and you sample a certain number of balls from the pool, you found then some number, possibly a small number, would be white balls, for example. And then the question you have is, what is the proportion of white balls in this huge pool? What is its distribution? What is the distribution of the log proportion? That is what we're trying to answer here with the Bayesian inference. In this case, we compute the expectation value of log expression with the posterior distribution, which is a beta distribution. At the same time, we also extracted cellular summary covariates to account for potential technical confounding. It has been known that sequencing depth can be a factor that confounds the UMI reads. So we have considered things like what is the log total UMI count per cell, and also the number of zero read genes per cell as covariates that confound expression. However, we also considered the nonlinear terms like the squared, for example, to account for the co-expression, to account for the expression confounding. And then these summary covariates can be integrated or aggregated with the existing covariates like batch effects. And all these covariates are regarded to change expression levels at mean and also log variance levels. Therefore, we computed their contribution to the mean and log variance and removed their effects, which ended up with normalized expression and also normalized covariates. After that, the normalized expression and covariates can be used for downstream analysis. For example, linear association test. In this case, we use a very simple linear model, which is essentially a conditional Pearson correlation. So Y would be the gene expression you want to test after normalization, and then X would depend on the scenario. So for differential expression, it would be binary 0-1 grouping, and for co-expression, it would be expression of another gene. In the end, you are testing whether alpha is 0 with exact likelihood ratio test for p-values, and also alpha would correspond to the log fold change. The advantage of the whole thing is, one major advantage is completely non-parametric, including the linear association test, and also the normalization part before that. So that you don't need to worry about any parameters in the whole process. We look at the question of differential expression and try to benchmark it with existing methods. So in this case, we randomly split the same group of cells into two for multiple time and see whether you find any gene differentially expressed while you're not supposed to. Well, that is indeed the case when you look at p-values, differential expression p-values from Normalisr, also from a recent normalization method, which is SE transform. So here we have a histogram of now p-values for genes separately considered from low to high expression based on the proportion of cells expressing that gene. However, for mass and agile, which were benchmarked as the best differential expression methods, they actually have biased null p-values. For example, mass is always slightly overconserved, and agile have a high false positive rate or high false negative rate, depending on the expression from low to high. So we then look at the question about positive control, or in other words, sensitivity in differential expression, especially between Normalisr and SE transform. In this case, we use, as before, we use the CRISPR-I perturb-6 screen data to see whether the target gene really had a differential expression. Normalisr was about as good as agile. However, for SE transform, you find really a reduced sensitivity, suggesting it finds a weaker signal in terms of p-value. And therefore, in this sense, Normalisr performed best in differential expression tasks. We also we also evaluated runtime, indicating Normalisr is thousands or even tens of thousands times faster than mass and agile, indicating it can be scaled to millions of cells or even more. We then look at the question co-expression. So again, we use the synthetic data set without any co-expression, and Normalisr could achieve really very good performance on the null p-value distribution, which is very close to uniform. On the other hand, if you look at SE transform, it had pretty biased p-values, is getting a very high false positive and some false negative, distorted null p-value distribution in general. At the same time, we also look at the reproducibility of co-expression, because we don't really have a ground truth of co-expression. We only see whether the co-expression were reproducible. In this case, we look at the 10 separate batches of the perturb-6 data sets, looking for co-expressions for each of the batch and see how much they overlap. In general, Normalisr had a much higher overlap rate than the methods, including SE transform, especially for the most significant co-expressions it found. And the overlap or reproducibility rate is actually pretty high. It's over 50% for about the top 1,000 genes. Given the small data set, in this case, we only chose the cells without any guide RNA for the co-expression. It's actually just about 400 cells for each batch. This indicates a very strong overlap and actually very strong significance. At the same time, we can also see how the imputation and normalization method really recover the ground truth of the expression levels of the biological variations, but not actually removing the technical variation. In this case, we compute the Pearson correlation with the ground truth on the synthetic data, because that is the only way we can know the ground truth. We found normalizers actually consistently performed on top regardless of expression. Log CPM actually was second, and SC transform couldn't compare with normalizer in this case. We then move to look at some real data set and see what kind of biology we could recover. In this case, we use a dysfunctional T cell activity in human melanoma. We first perform differential expression between dysfunctional and naive T cells, and on the left, we could recover the very well-known dysfunctional genes as annotated here. They are upregulated. We also look at what are the known dysfunctional gene sets as annotated in the previous data set, and we found significant enrichment of the annotated dysfunctional gene sets as well. This means normalizer could recover the known biology with differential expression. Then we move on to look at single-cell co-expression. So in this case, as you can see here, this is the co-expression we discovered. You don't really need to look into the details, but it's more or less like a hairball. But if here you have a lot of ribosomal genes indicating actually housekeeping roles might actually be dominating single-cell co-expression. And when we did GO enrichment on the top principal genes, meaning those with the most edges, we found this was indeed the case. It was the top GOs were mostly ribosomal subunit and so on, indicating translation activity. Well, these are definitely genuine co-expressions, and people may be interested in that, the translations in dysfunctional T cells. But personally for me, I'm looking into some of the more cell-type specific functions and genes in the co-expression. To do that, we'd like to remove these genuine but generic co-expression patterns by using principal component analysis. So in this case, as we just discovered, the top GOs were for the translation activity. So what we did was we look at the top one actually GO category and see what genes were included in this GO. And then we perform principal components on all these genes and added the first principal component as a new covariate. In this case, we have one more new covariate. We could do the whole analysis over again to find, okay, what's the new top enriched GO? In this case, the whole process would repeat and repeat over and over again, and it's only when the top GO is more cell type specific, like here responds to virus. So the only manual input in this process is the principal gene count and when to stop. So you generally don't need to worry about, well, how artificial the process is. And we found actually this is pretty reproducible across data sets and even cell types. So in this case, we added GO categories like cytosolic part, reflecting translation, and chromosome condensation reflecting cell cycle. So in the end, we obtain a co-expression network like this. So on top, you have two separate clusters indicating type 1 interferon response and cell cycle. And other than that, we annotated each gene according to their known function. And as you can see here, these are reflected in the outer edge color. Those genes are really clustered pretty well according to their known function, as you can see here. And we also have the log fold changes comparing dysfunctional T cells versus naive T cells. The edge color reflects whether the genes are positive or negatively co-expressed. So here we found genes sharing a similar function actually cluster really well on this co-expression network. And therefore, if you, for example, you have a gene of unknown function, you could probably infer its function from just its neighboring genes in this case. We also did more systematic evaluation of the co-expression network by looking at the overabundance of edges between genes in the same GO or CAG pathway. We found multiple significant overabundances encompassing cell type specific and more generic pathways, indicating the co-expression patterns are really reflecting genuine biology. Another case, we look at, well, whether we could infer gene regulations with a pooled high MOI CRISPR single cell RNA-seq screen. In this case, you have a number of cells and you have a pooled infection with a very high MOI. So each cell can contain, for example, 20 guide RNAs in average. And then when you do single cell RNA sequencing, you sequence the guide RNAs and also the transcriptomic profile, so you can infer how the transcriptome are affected by each of the guide RNA. So these, therefore, these can be very highly efficient screening systems. However, it can be subject to like potential false positives because as you know, guide RNA really compete a lot for things like the dCas9 protein, also the sequencing as well. But it has been unclear whether or how much they would give rise to false positive. So first of all, we use a really large two-day enhancer screen data set of over 200,000 cells to test this phenomenon. The hypothetical scenario is, although you have an effective guide RNA that inhibit the targeted gene, which consequently may affect other genes, completely unrelated guide RNA that doesn't do anything other than competing with this guide RNA. Here, the effective guide RNA here, the competition will give rise to false positives in the association between the unrelated guide RNA and other genes. In this case, the first question is, of course, whether we find the negative association or the competition between the guide RNA, and that is indeed the case when we draw a hypergeometric test on the guide RNA pairs. In this case, we find when you have a guide RNA in the cell, it's less likely for it to have another guide RNA. And then the second question is, we had this competition in place, would that give rise to false positives? And that is indeed the case. As you can see here, we perform differential expression on the guide RNA you want to test, but to ignore other untested guide RNAs, you have a very elevated distribution of the null p-values, indicating a high number of false positives. Actually, you have more false positives among the guide RNAs that are directly targeted by for the genes that are directly targeted by guide RNAs and their transferrin start sites versus other genes, indicating that these false positives are mediated by the other guide RNAs. Therefore, we could actually account for the competition by regarding every other guide RNA as a potential covariate. In this case, we would have lots of covariates, but we were successfully able to reduce the number of false positives into, for example, about 1.5% in terms of false positive rate. Therefore, with the competition-aware method, we could look for the guide RNA gene associations in terms of direct inhibition of the targeted gene, also in terms of the transassociation between the guide RNA and the distant gene. In this case, if we find association for both of them, we could use them to infer gene regulations from the target gene and the transgene. In this case, we would also like to remove the mediation by nearby genes, so that the guide RNA only affects or inhibits the target gene within neighboring 1 mega base pair region. At the same time, we want to remove potential off-target effects, also improve the robustness or reproducibility by looking for the transassociations that exist for the both guide RNA targeting the same gene, also in both the full scale and also the small scale screens. In this case, we ended with a list of gene regulations, but the numbers of targets for each regulator actually differs a lot. As you can see here, these are the top regulators we found, and many of them show very strong difference or preference in terms of activation or inhibition roles. We also did validation on their targets with the geo enrichments matching their known function. At the same time, for DDX3X, which is known to be responsible for mRNA processing, we also validated its targets with ENCODE shRNA knockdown experiment. We found agreement in the, for example, Pearson correlation. To conclude, here we propose Normalisr, which is a normalization method and framework that unifies single-cell differential and co-expression with linear association testing. So here, Normalisr achieved optimal specificity, sensitivity, and speed robustness in both of these tests compared to existing methods. It could also reveal really cell-type specific biology, especially at high-quality gene level for the co-expression networks. I would think co-expression networks would be an interesting study for many single-cell RNA-seq datasets. For CRISPR-I single-cell RNA-seq screens, actually we found very strong false positives from guide RNA competition, but Normalisr could account for that and reduce the false positives. It could give rise to really robust gene regulations inferred and recover interesting biology as well. In the future, we hope to extend Normalisr to more complicated studies like EQTL and causal inference at cohort levels. At the time of speaking, we are preparing our Normalisr package and also the preprint. So hopefully by the time you watch this video, they will be available on GitHub and also on bioRxiv. I would like to thank the people who contributed to the work or helped me during the process from within or out of the lab. I also have a complimentary poster for this talk. Thank you.